The slides are not coming, so we are going to yes. We have panelist one, Dr. Tunusri Powell, Phase A Year 2 resident, and panelist two, Dr. Sazat Hussein, Phase B Year 3 resident, and resident chairperson, Dr. Tasneem Binti Ahmad, Phase B Year 4 resident. And according to the program schedule, we will start first with the main presentation, after which there will be a question answer session, and audiences are invited to ask questions. Then we have short speeches from our panelists, one and two, and chairperson. And in the second part of the program, we have session from our German madam, Dr. Firdousi Begum, associate professor, and teacher session. I would like to proceed to the main presentation, and I will request Dr. Amina Begum to present her part of the presentation. Over to you, Dr. Amina.
Honorable Chairman, respected teachers, and my dear fellow colleagues, Assalamu alaikum. Me, Dr. Amina Begum, Year 1, Phase A resident in Department of
Volume is very low. Your volume is very low. Sound is very low.
Honorable teachers, Honorable Chairman, respected teachers, and my dear fellow colleagues, Assalamu alaikum. Me, Dr. Amina Begum, Year 1, Phase A resident in Pathology Department of BSMMU. I welcome you all to our today's journal club presentation on Mechanism of Disease, Autophagy in Human Health and Disease. At first, introduction. During the past decade, interest in defining the basic cellular mechanism of autophagy and its roles in human health and disease has become widespread. Autophagy is a homeostatic process, involves the sequestration of cytoplasmic components in double-membrane autophagosomes. The figure shows the phases of autophagic pathway. It proceeds through several steps. At first, formation of an isolation membrane or phagophore that is derived from endoplasmic reticulum, followed by vesicle elongation that captures the cytoplasm. Then maturation of autophagosome. This autophagosome then binds with lysosome and forms autophagolysosome. The content of autophagosome then degraded by acid hydrolysis of lysosome. The degraded contents then released for metabolic recycling. This figure shows the regulation of autophagic pathway. Autophagic pathway responds to input from environmental foods. The regulation pathway depends on mTOR, that is mammalian target of rapamycin. Starvation and energy depletion initiates the process of autophagy through inhibition of mTOR. Energy depletion also causes activation of UNC51 like kinase 1 that depends on AMP level. On other hand, nutrient signal and in response to insulin and other growth factor signaling, class 1 phosphatidyl inositol 3 kinase AKT pathway activates mTOR, thereby negatively regulates autophagy. Backlin 1 also negatively regulates autophagy through AKT, BCL2, BCL, XL, and uh, AKT and BCL2, BCL, XL mediated inhibition. There, there are also two conjugation systems that play important role in autophagy. These are ATG5 ATG12 conjugation system and LC3 ATG8 conjugation system. These are two ubiquitin-like conjugation system, and they regulate autophagy through elongation and maturation process. Now the functions of autophagy. Autophagy is a survival mechanism under conditions of stress. Turnover of mitochondria, that is mitophagy, and other organelles such as endoplasmic reticulum and peroxisomes. Clearance of polyubiquitinated protein aggregates. Regulator of lipid metabolism, protective mechanism that may prevent cell death. During infection, degradation of intracellular bacteria and viruses. Autophagy also participates in adaptive immune response through antigen presentation and lymphocyte development. This figure shows at a glance the effects of autophagy on disease progression. The beneficial roles of autophagy are
Louder, please, Dr. Amina. Louder.
beneficial roles are shown in blue color and harmful roles are shown in red color. Here, beneficial roles such as self-survival, energy maintenance, and metabolite generation. In case of cancer, cell death, chemosensitization, and tumor suppression. In case of vascular disease, plaque stabilization and regulation of proliferation. In case of infectious disease, antigen presentation, pathogen removal, and regulation of inflammation. And in neurodegenerative disease and aging,
Louder, please. Audience cannot hear you properly.
aggregate clearance and mitochondrial preservation. And harmful roles such as chemoresistance, radioresistance, and tumor growth, apoptosis, viral replication, etc. Now, I would like to hand over the microphone to my dear colleagues, Dr. Eshona Paul, to continue the rest of the session. Thank you all.
Thank you, Dr. Amina Begum. Am I audible?
Yes, please proceed. Please maintain that level of volume so that we can hear you properly.
Okay, sir. Thank you. Honorable Chairperson, respected teachers, my dear colleagues, Assalamu alaikum and very good morning to you all. I am Dr. Eshona Pal, Year 1, Phase A resident. Welcome you all in this session. Autophagy in disease, cancer. Monoallelic disruption of chromosome occurs in cancer. There is altered expression of other autophagy proteins. Chromosomal aberrations are seen in autophagy genes. It has an anti-carcinogenic function and tumor promoting effect also. Autophagy induces chemoresistance of tumors. Neurodegenerative diseases. Huntington's disease. Mutant huntingtin protein causing the autophagic pathway inefficient. Alzheimer's disease. There is accumulation of microtubule associated protein tau and beta amyloid peptide A beta. Mutations in mitophagy regulating protein PINK1 and PARK2 are seen in Parkinson's disease. Infectious disease. There are several human pathogens which are degraded by xenophagy, like bacteria, group A Streptococcus, Mycobacterium tuberculosis, Shigella flexneri, Salmonella enterica, Listeria monocytogenes, Frankicella tularensis. Viruses, herpes simplex virus type 1, chikungunya virus. Parasites include Toxoplasma gondii. Vitamin D and sirolimus. They inhibit the autophagy-dependent HIV and Mycobacterium tuberculosis replication. Adaptive immune responses leads to vaccine development. Single nucleotide polymorphisms are associated with Crohn's disease and ulcerative colitis, Mycobacterium leprae infection, and Mycobacterium tuberculosis infection also. There are different cardiovascular diseases which have modulations in autophagy, like cardiomyopathies, cardiac hypertrophy, ischemic heart disease, heart failure, ischemia, reperfusion injury, etc. Metabolic diseases, lipid metabolism and storage, Paget's disease, and autophagy is increased in different tissues like cardiac and skeletal muscle, adipose tissue, and pancreatic beta cells during exercise. Hepatic autophagy is downregulated in obesity and insulin resistance in mouse models. Now the pulmonary disease. There is increased autophagy in COPD, impaired aggriphagy in cystic fibrosis, and impaired autophagy in human idiopathic pulmonary fibrosis. Single nucleotide polymorphisms are associated with risk of childhood and adult asthma. Now, what is the role of autophagy in aging? During aging, there is accumulation of unprocessed material and autophagy declines. Downregulation of autophagy genes with age. Caloric restriction reverses the age-dependent decline in cyanoradicals in clinical applications. Sirolimus, that is an immunosuppressive and anti-cancer drug. It works by activation of autophagy. Chloroquine and hydroxychloroquine has chemotherapeutic efficacy. What is the take-home message? Autophagy can be both harmful and beneficial, widely implicated in physiological responses and pathophysiological processes. It can prevent pathogenesis, may lead to the identification of new targets for both diagnostic and therapeutic approaches. Thank you, audience, for your patience hearing.
Where is Dr. Kashim Haji? What will be your next session?
Madam. Sorry.
You continue.
Yes. Thank you, Dr. Amina Begum and Dr. Ishana Baul for your nice presentation. Now I would like to invite questions from the audiences. You can send it by chat box or unmute yourself and ask directly. We have questions in chat box. Dr. Sadia is asking about consequences of mutation in autophagy regulation gene. Dr. Amina and Dr. Ishana, I think you hear the question.
Thank you for the nice question. Thank you for the nice. mutation of
And
autophagy
speak loudly. We can't hear you.
as a consequence of mutation of autophagic gene, uh, some autophagic proteins or 5 autophagic gene is mutated. There is AG7 site or tumor progression, genomic instability, chance of
The sound is sound is broken. Sound is broken.
tumorigenesis.
Dr. Amina, would you kindly repeat? Repeat your
Thank you,
answer.
madam. Thank you, madam. Gene can result in tumor progression and the progression damage
Please adjust the volume first. Is it- Please adjust the
Please
volume.
repeat the question. Repeat the question
Yes, Dr.
and
Shadia,
give the
Madam,
answer again. We couldn't
Dr.
hear
Shadia
anything.
is asking about the consequences of mutation in autophagy regulating gene.
some. Thank you for the nice question. Uh, there are some autophagic genes such as ATG4, 5, Declin1, ATG7. This can be mutated. This can be mutated, and mutation of autophagic gene results in tumor progression and genetic damage and genomic instability. Thus, there's a role in uh, tumor formation, that is tumorigenesis.
Yes, we have another question from Dr. Tasmia Trisha. What is the role of autophagy in Alzheimer's disease?
Would you repeat, please?
Dr. Kashim, you repeat the question.
Ji, madam. Dr. Tasmia Trisha is asking, what is the role of autophagy in Alzheimer's disease?
Okay. Thank you, Dr. Tasmia, for the nice question. Am I audible?
Answer question. We can hear you.
Okay, ma'am. Yes, Alzheimer's disease involves the accumulation of the hyperphosphorylated form of microtubule associated protein tau. And there is also another accumulation, that is the beta amyloid peptide. And this beta amyloid peptide is named A beta. It impairs the lysosomal function. That means the autophagolysosome formation is impaired, and so there is impaired agryphagy. Impaired clearance of this microtubule associated protein tau leads to the formation of neurofibrillary tangles. This is the basic mechanism of Alzheimer's disease, which is related to autophagy.
Okay, next question is from Dr. Naimul Haq. He's asking, what are the cancers associated with autophagy?
I would like to Dr. Amena to answer the question.
Thank you for the question. There are some cancers which are related or associated with autophagy, and these are breast carcinoma, ovarian carcinoma, prostate carcinoma, due to monoallelic disruption of BECLIN-1 gene. And there is other carcinomas such as colon cancer and lungs and brain cancer is also associated with autophagy by mutation of PARK2 gene. Thank you.
Another question from Dr. Shukriya is asking, is there any role of autophagy in the vascular disease?
Autophagy has some roles in vascular disease, such as uh, in autophagy, the autophagy cause stabilization of atherosclerotic plaque and also regulates its proliferation. And does it play a role in vascular disease, which is protective?
Okay. Another question from Dr. Saida Siraju Mahmouda. She's asking how vitamin D is inhibiting autophagy dependent HIV and tuberculosis replication.
Thank you for the nice question. Here in this article, the author reviews that the vitamin D inhibit the replication of HIV and Mycobacterium tuberculosis by autophagy dependent mechanism. But the details mechanism is not included in this article. It is a review article, so just the decisions made from other articles are included here.
Okay, the question and answer session will end here, and we are going to the next panelist session. So we call now our panelist one, Dr. Tanusri Powell, to say her part.
Assalamu alaikum. Good morning to my respected teachers and my colleagues. Thanks, Dr. Ishana and Amina, for your nice presentation. Okay, autophagy is a ubiquitous cellular process responsible for the bulk degradation of cytoplasmic component through an autophagosomal lysosomal pathway. Genetic screen of Saccharomyces cerevisiae, a yeast, have identified numerous genes that are essential for autophagy. Many of these genes have orthologs in higher eukaryotes, including Cyanorhabditis elegans, Drosophila, and mammals. So so now, as they have already discussed about the effect of autophagy in human health and disease, so I would like to focus some light on the effect of autophagy or function of autophagy on those animals. Here, different journals and articles publish that the major known physiological function of autophagy in multicellular organism, largely determined by knockout study in mice, include provision of nutrient during catabolism, generation of ATP in stressed or starved cells, generation of signals for heterophagic removal of apoptotic corpses, degradation of misfolded protein, damaged organelles, and preservation of genomic stability. These are the physiological functions they have found on those animals. So the main function they have included the lifespan extension on these animals by mutation in insulin-like growth factor 1 or factor DAF2 or and factor AGE1. This mutation is helpful in increase in adult longevity in these animals, and they also included dietary restriction plays an important role in lifespan extension in yeast, flies, and mammals. Autophagy also helps in survival during starvation in these animals, and the survival is done by when they are starved. Autophagy is activated in the pharyngeal muscles. Autophagy is also helpful in their reproductive development, and this is done by BAKE1 activity, which is required for viability and fertility of these animals. Then autophagy in synapse development. Basal levels of autophagy are required for the survival of both neurons, and the specific impairment of autophagic degradation in neurons result in axon degradation, neuronal cell death. Now, autophagy in cell fate specification by microRNA mediated gene silencing in these animals also have been shown. Autophagy is also helps in stem cell proliferation. Autophagy promote proliferation of stem cell germline progenitors. So it directly or indirectly promotes cell cycle progression of mitotic cells in the germline. Autophagy is also important in cell size regulation, because when there is a scarcity of food or environmental stress, then the cell growth is suppressed, so the cell can survive. So on understanding the role of autophagy as an essential process in Cyanurhabditis elegans development has advanced greatly. So we have learned that autophagy is involved in cell survival, normal development, negative control of cell growth, longevity, clearance of toxic aggregates, the proteins, and cell surface receptor trafficking. Unlike humans, there is no talk about tumorigenesis in animals. So despite the great importance of autophagy in the physiology of higher eukaryotes, the molecular mechanism and functions of autophagy in this organism remains vague. So hopefully, Cyanurhabditis elegans, a genetically tractable system, will continue to allow us unravel the mechanism of autophagy regulation, execution, and function at the molecular and organism level further. Thank you.
Thank you, Dr. Tunusri Paul, for your part. Now I will proceed to our panelist two, Dr. Sazzad Hussain.
Thank you, Dr. Kasim Hagi. Honorable Chairman, Madam, respected teachers, and my fellow colleagues, Assalamu alaikum. We have known that the role of autophagy have been very important for imparting its role in the tumor suppression and various immune and inflammatory response. I would like to give some light over histopathology changes and tissue-specific response related to autophagy in various organs. Though the changes are not in humans, various studies have gone through various animal models. If we can see the changes in the liver, we can assume that it has very important role in lipid metabolism and lipid storage. And there are just some specific pathological changes like sinusoidal dilatation, cloudy swelling, or injury, and periportal and intraparenchymal inflammation, and various others. And the increased autophagy results in increased amount of autophagolysosomes, and the lysosomal granule and enzymes in the lysosomal enzymes causes various tissue damage. Like in case of cardiac myocytes, we can see that there is a myocyte degeneration because of increased autophagy. And there's some excessive autophagy may also causes some changes in nucleus. There's some specific nuclear features like there's a nuclear concavity and adjacent ballooning of the perinuclear shape, and nuclear membrane convolutions, and mild chromatic condensation. And there's also dilated and fragmented endoplasmic reticulum, and the separation of inner and outer nuclear membrane are also seen. And there's a high level of autophagy can also lead to autophagy gene-dependent and caspase-dependent cell death. And I'd like to focus on more thing that the excessive autophagy can also include some vesicle nucleation, vesicle elongation, and vesicle completion, which are regulated by various autophagy-related proteins. Lastly, I can say that there's sometimes autophagy pathway is uniquely capable of degrading the entire organelles like mitochondria, peroxisome, and endoplasmic reticulum, as well as intracellular organisms also. Then consequently, we can see the changes and reflects the changes over the tissue samples. So this is all from my part. Thank you. Thank you all.
Thank you, Dr. Sazzat Hussain, for your part. And now I would like to call Dr. Tasneem Binti Ahmad, our resident chairperson.
Assalamu alaikum. Am I audible?
Yes, we can hear you. Proceed.
Thank you. Respected teachers and dear colleagues, good noon. Today I would like to talk about Nobel laureate, Mr. Yoshinori Osumi, a Japanese cell biologist who received Nobel Prize in 2016 for his work or discoveries of mechanism for autophagy, which is related to today's topic also. Mr. Yoshinori Osumi, he, after starting his work own lab in 1988, he focused his all interest to the field of autophagy. And before that, in early 60s, 1960s, the concept of autophagies were emerged, and the researchers observed that the cell degrades its own components by forming membrane-bound vesicles. And Mr. Yoshinori Osumi focused his interest and in this membrane-bound vesicle, which degrade the cell components. And at first he used Baker's yeast. That means he used yeast for his research work, and he exposed some chemicals to this yeast. These chemicals produce some random mutation, and this mutated yeast produce some vesicles without vacuoles that does not contain any degrading enzymes. At the simultaneously, he also stimulated the yeast cell by starving, and within hours, he observed that the vacuoles are accumulated by small vesicles, which are not actually degraded. Then he find the autophagosome, or these vacuoles, which does not degrade the cell products. He find the autophagosome in the yeast. And then he, within 1 year, he also identified the gene which is related to this autophagosome, the key gene that maintain the autophagosome for doing its work. And within subsequent years, he also find the protein complexes which are encoded by this gene, and also show that the mechanism of autophagy, every steps of the autophagy procedure are controlled by the cascade of protein and protein complexes. And also he also showed that same identical mechanism also present in human bodies, human cells. And in 1992, he published his work and make the world know how autophagy works and how cell recycle its own content. And then he also opened a new path to understand the importance of autophagy, which is related to several conditions in physiology as well as in medicine. In physiology, like starvation, adaptation in starvation or response to infection, and in the pathological condition like cancer and several neurodegenerative disease. And he also opened a new field for researchers to do work on ground of autophagy. And for his this work, he was awarded Nobel Prize. And that's all from me. Thank you.
Thank you, Dr. Tasneem Ahmad, for your contribution. Now I would like to proceed to our teachers session, and our respective teachers can ask questions and add their comments regarding today's presentation.
Yeah, thank you, Dr. Amina and Dr. Reshona Pal, for your presentation. Is there any question from teachers?
Yes, madam. We have a question from Dr. Saika Habib, madam. Uh, what is the antiviral role of autophagy in herpes simplex virus?
Dr. Amina or Dr. Ashona Pal? Answer these questions. This is asked by Dr. Saika, madam. What is the antiviral role of autophagy in herpes simplex virus? Dr. Amina?
Thank you, madam, for this important question. Um, in herpes simplex virus infection, there is major histocompatibility complex class 2 antigen presentation to CD4 T cells. And if it is inhibited by the knockdown of ATG12 and ATG5, then the infection occurs in a greater rate. This is the relationship between the herpes simplex virus infection and autophagy.
I have a question. Can I ask, madam?
Of course.
I have a question to the presenter and all the panelists, with a special attention to the supervisor of this presentation. Usually autophagy, what we used to heard during the month of Ramadan, the fasting, especially the religious fasting, can help us reducing the bad cholesterol from our body by the autophagy process. But the exact mechanism, or is it beneficiary for our body, I don't understand. Would you please clear it by any explanation or mechanism, how it is reduced our bad cholesterol from our body?
Dr. Amina, do you want to answer this question? Amina?
Japanese cell biologist Nishimori Oshumi also
We can't hear you. Speak loudly or correct your microphone or something.
Japanese cell biologist Yoshimori Oshumi also established the connection between autophagy and fasting. This established connection is that fasting increased blood sugar level, it reduced inflammation, caused weight loss, and it improved brain function. These are the connection between fasting and autophagy.
Thank you, Dr. Amina. I'd like to add to your answer. Thank you, Dr. Vishnu. And also you mentioned me to give feedback as well. That's why I am adding to the answer to Dr. Amina Begum. And the thing is that these fasting related benefits in autophagy is not yet established in human. It is established in Cyanorhabditis elegans, there is a worm, and some mouse models. So in those mouse models, it shows some beneficial effect, but only after the fasting continues for 72 hours straight in mouse models. In the meantime, the animal had to remain hydrated, but without water, nothing else was given. So then the autophagy kicks in after about 72 hours. But since human has a much lower metabolic rate than rodents or worms, it is likely that to trigger starvation associated autophagy in humans, we need to starve a human for about 30 days, which is unethical. So it has not been experimented so far I know. But so starvation associated benefits of autophagy in humans are not well characterized, and but in other roles, like its roles in different cancer, both beneficial and harmful, and different neurodegenerative diseases and etc., these roles are has got clinical implications in humans. But as far as I know, the starvation has been studied in non-human models because it is not practical to study it in humans. Thank you.
Kabir bhai, is there any question from you or comment? Kabir bhai, can you hear me? Kabir sir, are you here? Okay, Vishnu, do you have any question or comment or?
Yes, I can hear. I have a question about Mycobacterium tuberculosis and autophagy. Can you clear?
Yes, sir, we can hear you, sir.
What is the relation of autophagy and Mycobacterium tuberculosis?
Dr. Amina or Dr. Eshona? Who wants to answer this question? What is the relationship between Mycobacterium tuberculosis with this autophagy?
First, unmute yourself. Unmute.
Dr. Amina, would you kindly unmute?
Thank you, madam. In this article, authors shows that if macrophage-specific deletion of ATG5 occurs, then increase the tuberculosis susceptibility.
That means what it will do?
That
Is autophagy prevent infection or?
Yes, madam.
It prevent infection. Dr. Soumitra, would you like to supplement her?
Yes, madam. Actually, the autophagy is a very complex process, and some of its gene seems to confer advantages against different infections and certain neurodegenerative processes and associated diseases. And so far, this knockout models has been studied in different animals because we cannot ethically knock out genes from humans. Humans has got orthologous or homologous genes, but thus far no clinical case has been identified. But since the animal models suggest it, we think it could be clinically important or therapeutically important if those genes are targeted. But we have to keep in mind that autophagy related particular genes related to autophagy are associated with different clinical targets, but autophagy as a process as a whole, uh, 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 it confers some advantage and disadvantage which may or may not be associated with the function of a particular gene in that pathway, because autophagy can act in multiple ways. Thank you.
If anybody, any comments or question? Okay, at last I'd like to thanks again Dr. Amina and Dr. Eshona Paul for their presentation and other panelists. And thank you, everybody. So I think we can conclude our session. Okay, thank you for participation.